Hello students, today we are going to see the next type of cell in the form component, that is WBC. So let's just start with the WBCs. What is the WBC? That is white blood cell or leukocyte. Okay. So first thing we should know that in per cubic millimeter of blood, how many WBCs are present, right? So first thing that the WBC count, okay, the total WBC count is known as TLC, total leukocyte count, okay. So that total leukocyte count is around 6,000 to 8,000 WBCs per cubic millimeter. Okay, next thing you need to know that this WBC is actually going to have around 12 to 20 micron in shape. Okay, the diameter will be around 12 to 20 micron. The next characteristics of the WBC, that this WBC are going to be a cell which have irregular surface area. That means it is not going to have a definite shape and structure. Okay, so it is irregular in shape. Okay, so this is one important point, that WBCs are irregular in shape. The next thing, that in case of the WBCs will have all the organelles, that it is going to have the mitochondria, it is going to have the nucleus, it is going to have endoplasmic reticulum, Golgi apparatus, everything that will be present in the WBC. Okay, so that's why the WBC will actually can undergo rapid divisions also. Okay, so this WBC have nucleus, mitochondria. So WBC will have nucleus, mitochondria, endoplasmic reticulum, Golgi body, Golgi apparatus, everything along with other organelles that will be all intact in the WBC, which have irregular surface area. Okay, so that is important. Another important point. Now we will see some conditions. Okay, so what happened if an individual, okay, if so, see the conditions. What are the conditions? If an individual increase TLC, that means the total leukocyte count from 6,000 to 8,000, if it is increased, okay, then this condition, this WBC, if it is increased, then what we can call it? This is known as leukemia. Okay, so you know what is leukemia. Leukemia is one form of cancer. Blood cancer is nothing but actually the leukemia. Okay, and if the WBC count is decreased than that of 6,000 to 8,000 WBC per cubic millimeter, it is known as leukocytopenia. Okay, fall in WBC count is known as leukocytopenia, and if the WBC count is increased, then it is known as leukemia. This WBC is going to have a very unique characteristics. What is that? Yes, it is present in the blood, okay, but it can squeeze out of the blood vessel. Okay, so it is not going to have its definite region where it is going to present. It will be present everywhere in your body. Okay, it can come out of the blood vessel, and there is a phenomenon by which the WBC can squeeze out of the blood vessel and can go to each and every region in the tissue spaces. Then what we can call it, that is known as diapedesis. Okay, so what is diapedesis? Diapedesis is a phenomenon by which the WBC can squeeze out of blood vessel. Okay, so WBC can squeeze out of the blood vessel and can go to each and every region, because this WBC have a chief function to fight against the pathogen. So anywhere we have the pathogen, so that pathogen can be killed by this WBC. So that WBC that have to go to any regions of the body. So that's why it is going to have irregular surface area, so that it can squeeze out of the space present in that blood vessel. So from this blood vessel, this WBC can squeeze out of it and it can go to any region. Okay, wherever we have infection, allergic reaction, suppose in that region, WBC will go, and that phenomenon is known as diabetes. Normally also it is possible that our WBC count can increase whenever our body have suppose some infection. Whenever we suffer from some disease, then the WBC count will normally increase, but abnormal secretion will be known as leukemia. That is actually one form of cancer, blood cancer. Now we will see the types of WBC. Okay, so before coming to the types of WBC, we need to know where it is actually formed. Like the RBC, this WBC will be also formed in the bone marrow. Okay, it is also formed in the bone marrow, and the different type of WBCs that will get matured, in different regions. Okay, that will get matured. If it is the B lymphocyte, it will be matured in the bone marrow, and if it is suppose in the lining of the alimentary canal, and if it is a T lymphocyte, it will going to get matured in the thymus. Okay, so let's just see the different types of WBC. So whenever it comes to the classification of WBC, then we are going to have two types of WBC basically. What are they? One is known as the granulocyte. Okay, and the second type of WBC, it is known as agranulocyte. Okay, now this granulocyte will be of three types. Okay, what are they? One is known as eosinophil. Then the second type of WBC is known as basophil, and the third type of WBC is known as neutrophil. Okay, and there are two types of agranulocyte. What are they? One is known as monocyte. Okay, and the second type of agranulocyte is known as lymphocyte. Okay, so just see, if it comes to the granulocyte, then in the cytoplasm there are some granules present. Okay, that will be known as the granulocyte. Okay, and if it is the agranulocyte, they are going to have clear cytoplasm where the granules are absent. Now after that, we need to know some characteristics of the WBCs also. Like if it comes to the eosinophil, basophil, neutrophil, they will perform different function. So let's just see what is it. Okay. Now just see, if it comes to the different types of WBC, then what we have seen that there are two types of WBCs, agranulocyte and granulocyte. In case of agranulocyte and granulocyte, once again we have divisions. What are they? These granulocytes, they are of three types: eosinophil, basophil and neutrophil. And agranulocytes, which have clear cytoplasm, are of two types. One is monocyte and other is lymphocyte. Now first of all, we need to know, and yes, this is very much important, that you need to know this chart, that classification is going to be very much important. Okay, so this classification is important. You have to know the different types of WBCs, their functions, everything. So whatever I'm writing here, that is going to be very much important. From here questions are asked. Okay, so what are the questions that can be asked? That in case of the WBCs, which one have the trilobed nucleus? Okay, so you have to remember, and that's why I have given this way synchronization, so that you can remember. What is that? If it comes to eosinophil, it is going to have bilobe nucleus. It is headphone shaped bilobe nucleus. If it is basophil, it is going to have trilobed nucleus, and if it is neutrophil, it is going to have multilobed nucleus, where the lobes can vary from 3 to 7. So you can remember this way: eosinophil, basophil, neutrophil, which have bilobe nucleus, trilobe nucleus and multilobe nucleus. Okay, otherwise if it is changed, it is very much difficult to understand. So just remember eosinophil, basophil, neutrophil, 2, 3, 3 to 7. Okay, so bilobe nucleus, trilobe nucleus and multilobed nucleus. So this is the first difference. Second thing, that in case of the monocyte and lymphocyte, monocyte is going to have bean-shaped or horseshoe-shaped nucleus, and if it is lymphocyte, it is going to have the round nucleus. Okay, so this is the first question. Second question is asked that in case of the WBCs, which is found in minimum amount and which is found in maximum amount. So first of all, we need to know their concentration also. If it is eosinophil, this is around 2 to 3% less. If it is basophil, yes, this is going to be very much important. It is found in very less amount. Okay, it is found in the least amount itself. What is that? 0 to 1%. And if it comes to the neutrophil, it is also very much important because it is found in highest amount, that is 60 to 65%. Okay, and if it is monocyte, around 2 to 10%. It is lymphocyte, around 20 to 25%. Okay, so that is also important. Next one, what is the function, right? The function is also going to be important. What is that? It is non-phagocytic. Now what is phagocytosis? Phagocytosis means, suppose there is a foreign particle comes, this WBC will engulf and remove it outside. Okay, so that is the phagocytosis in simple definition. Okay, so it will not engulf the foreign particle. It is not actually in phagocytic nature. It is non-phagocytic. Next one, it will help basically in which function? The function is allergy. Okay, remember, whenever we have allergy, then the eosinophil level will rise. Next one, if it comes to the basophil, remember I told about the mast cell in the connective tissue whenever we have discussed the alveolar connective tissue. So mast cell was releasing 3 chemicals, right? Histamine, heparin and serotonin. Same thing, that this basophil will also release the same thing, that is heparin, histamine and serotonin. I told like the mast cell in the alveolar connective tissue. I will not describe the characteristics of that heparin, histamine, etc. in now, because earlier video already I have discussed what is the function of heparin, histamine and serotonin. Now come to the last cell of the granulocyte, that is neutrophil, which is now phagocytic in nature. Okay, so it is phagocytic in nature, which is going to engulf the foreign particle and dead cell. If suppose there is a dead cell, whether it is the normal tissue cell, somatic cell or the RBC suppose, then this RBCs are going to engulf by this neutrophil and it will bring back to the spleen so that it can be destroyed. Okay, so that's why it will be phagocytic in nature. It will engulf the germs also, and it will actually phagocytose the dead cell also of our body itself. So these are the functions of the granulocytes. Now come to the monocyte. Monocyte, which is going to have this horseshoe or bean-shaped nucleus. Now it is motile in nature. It is also non-phagocytic. It will secrete the antibodies, okay, which is helping in healing. Antibodies means immunoglobulins. Antibodies, globulins, this is present in the plasma that is actually secreted by the monocyte. Remember, we are going to have 5 types of antibodies. All the functions of all the type of antibodies and their structure that will be needed in the second year, not now. So just remember now that in case of monocyte, it will basically help in secreting the antibodies, okay, which will fight against the pathogen or foreign particle. Next one, come to the lymphocyte. Lymphocyte is also phagocytic in nature. It will engulf the cell debris, and whenever cell debris means the excretory product of the cell, or if suppose some cells are died, then it will be killed by the lymphocyte, and that lymphocyte, if it comes out of the blood vessel and go to the tissue, it will convert into a macrophage. Okay, so what is macrophage, what are the antibodies, those whole immune system part will come under second year, that will be discussed in the next year. Now this is about the different type of WBCs. So these are the different types you need to know. After that, let's just move to the platelets. Now just come to the next third type of cell under the form component is the platelet, right? So this platelet is also known as the thrombocyte, which is also a nucleated cell. That means now in the mammalian blood there are two cells which are without nucleus. One is RBC and another is platelet. But other than mammal, the rest of the organism like fish, amphibia, reptilia and ape, they're going to have the nucleated RBC. Okay, remember that. Along with that, nucleated platelet too. Okay, so next, see the important characteristics of the platelet, that the platelet amount in our body is around 1,50,000 to 3,50,000 per cubic millimeter. It is highest present in the blood. Okay, next one, what is the structure shape? It is basically a cell fragment which is formed from a cell in the bone marrow called megakaryocyte, which is now oval in shape, round in shape, or it is disc-like also. Okay. Now the next important thing, that what is the lifespan of the platelet? It will live about 7 days. It is a cell which can survive for a week, and it will destroy in one organ, that is spleen. So spleen is the organ where the RBCs also died and also the platelets. Okay, it's not only the spleen, in the liver also the platelets die. Next one, if it comes to one condition when the platelets are going to decrease in the body, that will be known as the thrombocytopenia. And thrombocytopenia is a condition when the blood do not clot in the cut. Suppose one individual have a cut, then due to this less level of the platelets, the blood will not able to clot. So what is the main function of the platelet? It is the blood clotting mechanism. So this blood clotting mechanism, actually you should know in the next chapter. Another chapter we have, that is body fluid and circulation. In that chapter we are going to see in detail what is the blood coagulation mechanism, but little thing about the blood clotting mechanism in a very concise way I need to tell you, that whenever the blood, suppose the blood vessels are cut, then the platelets get injured, it releases a chemical that is known as thromboplastin, and that thromboplastin will actually stimulate our body to produce a enzyme that is known as thrombokinase. So that thrombokinase will convert the prothrombin, it's a component present in the blood, so that prothrombin will be converted into the thrombin in presence of the thrombokinase, in presence of also one ion, that is calcium ion. And that thrombin, that is the product that is formed in this reaction, that will act as a enzyme or catalyst for the next reaction, that this fibrinogen in presence of thrombin will be converted into insoluble fibrin. So whenever the blood vessels will cut, suppose these are the blood vessels that get cut, this fibrin will act as a reticulate structure so that the blood will clot. Okay, so this way the blood will get clot with this help of fibrin. Okay, so in presence of vitamin A and calcium ion, okay, so that fibrinogen will be converted into fibrin. So that is about the form component, that is platelet. What is the main function or the chief function of the platelet is to undergo blood clotting. There is also one genetic disorder when the blood clotting mechanism, the whole blood clotting process cannot be proceeded or cannot be done, that is known as, actually this is one condition, that individual cannot undergo blood clotting. Okay, so that is actually one genetic disorder, but whenever it comes to the platelet will undergo this mechanism to clot. Okay, so that is about the three types of cells. I have already discussed RBC in the earlier video, and in this video we have completed the WBC and the platelets. So that are the three types of foam component present in the blood. In the next video we are going to see once again the plasma part, what are the components present, and then the lymph, that is the next type of fluid connective tissue. So thank you. So that's all about the foam components. Hope you have understood. Thank you.